Welcome to your photosynthesis lecture. These PowerPoints can be found in your D2L contents folder for chapter 10. There we go. All right, awesome. Let's go ahead and begin. And so photosynthesis. We've already talked about photosynthesis when we introduced our cellular respiration. And so now we need to see the process that is going to make that sugar that is used in cellular respiration. So really, the process of photosynthesis is what feeds the entire biosphere. So if you recall in chapter 1, we talked about the biosphere. Sorry, I was looking at microphone permissions. I think we're good. So if you recall in chapter 1, the biosphere is the entire Earth. And so photosynthesis is a process that converts solar energy into chemical energy. So either directly or indirectly, photosynthesis is going to feed the entire world. So when we talk about directly, we're literally talking about the plants photosynthesizing, making their own sugar, and then they use that sugar within their own plant self. They send that sugar, i.e. glucose, to the mitochondria, and their mitochondria are going to produce the usable form of energy called ATP, and it's usable because it's available to do work. Indirectly, if you recall in the energy chapter, I made you I made you an ecosystem where the sun is going to uh, provide the energy for plants to photosynthesize. And then the bunny rabbit is going to eat that plant energy. He's going to take the sugars from the plant, and he's going to use those sugars, send the sugars to his cells, and those cells are going to transform that energy from the chemical energy of glucose into the chemical energy of ATP. And so that is a coupled reaction. When energy leaves the glucose, it is going to get uptaken by the ADP molecule in order to attach the covalently bond the P on, making ATP, which is a high energy molecule. So thereby it is feeding us because that ATP is available to do work. How's it feed us? I don't know, maybe you eat bunny rabbits. All righty, so photosynthesis is going to nourish the entire world. We have terms that you should know. Term autotroph. Autotroph, they sustain themselves without eating. So they are the producers, i.e. the plants are autotrophs. There are other autotrophs that aren't plants. And so some protists are autotrophs, and not all of them. And they're not only autotrophs, they usually do a combination of different nourishing. So they could be autotrophs, they could be heterotrophs, they could be a mix of the two. Also, they could be decomposers. And so just to kind of get you out of that thought process that plants are the only autotrophs, because that is not the case. But they are the main autotrophs, or I should say the most recognizable that in our viewpoint autotrophs. But all right, so autotrophs, like I said, are the producers of the biosphere. They produce that organic molecule from CO2 and other inorganic molecules. So H2O, CO2, they're going to come together with solar energy to produce glucose, which is C6H12O6. And if I'm not writing these out, these are because these are things I've already written out for you in the previous chapters. So almost all of your plants are going to be what is called photoautotrophs, okay? Photo meaning they use sunlight or some type of light. Autotrophs. They automatically make their own food. There are autotrophs called chemoautotrophs, meaning they don't get their energy from the sun, but they get their energy from chemicals. And those are the ones that we talked about that could live in the vents of geysers, in the deep sea vents. They actually obtain their energy from chemicals that come up from the earth. So nice little beautiful rainbow. What are you seeing? Reflection of light is what you are seeing. So photosynthesis occurs in plants, algae, and certain protists. So algae is a protist. So to separate that out is kind of redundant. And then also some prokaryotes. Interesting enough, the prokaryotes, these were your original photosynthesizers. These were your original organisms that came first before any other organisms on Earth. And so these organisms, all of the autotrophs, they not only feed themselves, like I was telling you before, but they feed other organisms through that food web. So here is your example of your plants. Here's your example of multicellular algae. Multicellular algae such as kelp or seaweed, if you want to say. And then you have unicellular protists. Unicellular protists are going to be example Euglena. They are microscopic unicellular protists. And then you also have cyanobacteria. So cyanobacteria, these right here are the organisms that literally changed the Earth's atmosphere from an inhospitable atmosphere that could not sustain life to the oxygen-rich atmosphere that we need now in order to sustain life. And then also you have your purple sulfur bacteria. So your purple sulfur bacteria, these are going to be your chemoautotrophs. So photoautotroph, 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 and photoautotroph, meaning these all use light as their energy source. So your heterotrophs. Your heterotrophs, they obtain their organic material from other organisms, meaning they do not make their own energy. What they do is they have to obtain their energy from another organism, either by eating, as such as an animal eats other organisms and gets its energy from them, or possibly from decomposing. Decomposing organisms such as fungi. Fungi are going to decompose other organisms and suck up, or they're going to, I say suck up, they're going to digest on the outside of their body, of their fungal bodies, and then they're going to bring in the nutrients in that fashion. So your heterotrophs, they're going to be the consumers of the biosphere, okay? So your autotrophs are the producers. So make sure you know those vocabularies. Autotroph, photoautotroph, chemoautotroph versus heterotroph, and then producers versus consumers. Almost all your heterotrophs, including human beings, they are going to depend on photoautotrophs, meaning you could get rid of all of the heterotrophs in the world and life will still continue because the autotrophs can make their own food. But if you get rid of all the autotrophs in the world, that's it, because there's a finite amount of food available for those heterotrophs. So your Earth's supply of fossil fuels was formed from the remains of dead organisms that died hundreds of millions of years ago. And so those fossil fuels, they represent stores of solar energy from the past, and very specifically, the Carboniferous period. So let's go ahead and see if we can find a nice little internet picture of that Carboniferous period. Let's see. I'm going to stop this share. Give me a moment. getting those perfect pictures out for you. Okay, so let's go ahead and share that screen. I went to the internet and found a nice, beautiful picture of the Carboniferous period. And what we're looking at here is we're looking at a representation of what plants look like millions of years ago. And so there was a high abundance of specific type of plants. And because of the so fern-like types of plants, lycophytes, if you go into 112, you'll get to learn a lot more about ferns and lycophytes and such. So because of the conditions at the time, very wet, very hot, lots of peat, very mossy. And so when these plants would fall over, when they would die, you can see as they die, they would get covered up in the bog. They would get covered up in an anaerobic type of an environment with lots of mud, lots of water and such. And it was a very anaerobic environment. So because of that, all of the carbon that is in these plants, they were not broken down. So normally through decomposition, that carbon would get returned to the earth. But because of the conditions of these plants, of the environment that these plants are living in, the carbon in the trees and in those plants and ferns and such was not able to be broken down completely. And so it was just partial decomposition. So that partial decomposition with lots of heat, lots of pressure, lots of time in that state of anaerobic environment led to the perfect conditions to lead to the making of our fossil fuels. So when I talk about our fossil fuels, we're talking about oil, we're talking about natural gas, coal. So the fossil fuels that we depend on today for energy to keep our house warm, to drive our car, to do all of those things, provide our electricity. Now, for the most part, not everybody is using clean energy right now, but all of those fossil fuels came mainly from the Carboniferous period, because that is when the perfect conditions were to build up those huge stores. So when those stores are gone, they're gone. And so that is why it is so important to try to find alternative fuels, and not only just alternative fuels because we're running low on them, but also alternative fuels because of clean energy. So those fossil fuels, they put out a lot of, oh, I just put my PowerPoint in the trash. Maybe not. There we go. Those fossil fuels, they put out a lot of dirty waste up into the atmosphere. So you get a lot more nitrogen, you get carbon dioxide, in some cases methane is released. And so they puts out a lot of unclean waste. Versus, if you look at this example right here, what we're looking at is we're looking at synthetic chloroplasts. Let's make sure I'm still sharing. Yes, I am. Okay, so these synthetic chloroplasts, they are basically doing the job of photosynthesis. So your chloroplasts are found within plant cells. And so within a plant cell, chloroplasts, they obtain the solar energy, and then through that solar energy, they will go ahead and process that energy and package that energy into a usable energy molecule, and that energy molecule is called glucose. Well, this right here, this picture that I'm showing you, this is a clean energy. We're using synthetic chloroplasts to do the job of plants. And as you can see, it is set up to a factory, and it says right here in the corner, green fuel. So lots of energy produced here. So your chloroplasts, they are structurally similar to and likely evolved from photosynthetic bacteria. And so the structural organization of those cells, that is what allows for the chemical reactions to happen in photosynthesis. So I'm going to go through a lot of vocabulary. I might scoot through these slides because sometimes I will just talk and you can go ahead and refer back to these slides. Vocabulary that we're going to cover is a chlorophyll, mesophyll, and then your cell is going to contain mesophyll. So what mesophyll is a tissue and it's made up of mesophyll cells. And the mesophyll cells, each mesophyll cell is going to contain about 30 to 40 chloroplasts. And it is in those chloroplasts that you are going to find the pigment chlorophyll. It's a green color pigment, and that pigment chlorophyll, that is what makes that chloroplast be able to sequester the solar energy. Other vocabulary you're going to want to know is stomata. Stomata or stoma, one is singular and one is plural, so this is singular. And so just think about O. Think about a hole, O. And so it's a pore, it's a hole in the leaf. So it is the hole in the leaf in which CO2 will enter into the leaf of the plant, and O2 is going to exit out of the leaf of the plant. And then so your chlorophyll, it's going to be found in the membranes of the thylakoids. We'll go ahead and talk about those pretty soon. And the term stroma, if you notice, these are two different. Stoma or stomata with an O and a T. Stroma has an R. So I always tell people, think about stroma with an R being the jelly in the room. Okay, so it is the interior fluid that is found within the chloroplast. So it's equivalent to cytoplasm, or it's equivalent to matrix, which is found in your mitochondria. All right, so let's go ahead and view. I just want to move my little thing up here. Give me just a moment. There we go. Perfect. What do you say we go to highlighter mode instead? There you go. Here's your leaf cross section. So if you look inside your leaf, so here's your leaf right here, and you do a little cross section of that leaf, it contains what is called mesophyll. That mesophyll is the tissue. So the leaf is the organ, the mesophyll is the tissue that makes up the organ. And then from there, each of these cells, each of these individual cells are called mesophyll cells. And so you can see they have little dots inside, little darker dots, and those little darker dots are called chloroplasts. So let's not go too deep into that just yet. Let's go ahead and look at the exterior of the leaf. Here's the exterior of the leaf, and you can see these little holes, those little holes being called stomata. Stomata is plural, stoma is singular. I think I might have said it backwards before. CO2 goes in and O2 comes out. So that is how your plant is going to obtain its CO2. And that is how it releases excess oxygen as waste. So these are veins. These are specific type of tubes that are going to bring water to parts of the leaves. And then there are other specific venules that are going to remove sugar from the leaves and take it to the rest. So you may have heard of these before. When the veins are given a specific name, they're called phloem and xylem. You'll hear more of that when you take 112. So if you look here, here's your mesophyll cell. All of these are mesophyll cells. We look inside our mesophyll cell, okay? You see all of these dots. All of those dots are called chloroplasts. It's time to go to a pen. So each of these dots, I did not mean to do red. Sorry about that. Let's try again. Pen color black. There we go. All righty. So each of these dots, boom, those are organelles in a cell, okay? And those organelles within the cell are called chloroplasts, okay? So there's your chloroplast right there. And if you were to go ahead and look inside the chloroplast, this is what the structure looks like inside the chloroplast. So first thing, let's go ahead and look at the anatomy of the chloroplast. So your chloroplast has two membranes. It's got an outer membrane, you can see that here, and it's got an inner membrane. So, and between the two, it has something called intermembrane space. It's basically space between the two membranes. Within the chloroplast, on the interior of it, you have these structures called thylakoids. And so these thylakoids, so if you look here, one of these, I feel like we need to get a different color other than yellow, and it is not providing that. Let's see. Yeah. Probably gray might be the best. Maybe. No, gray was awful. Give me just a moment. Working with my little mouse pad here. Let's go with black. Okay. So one of these thylakoids, boom, looks like a poker chip, right? So think about each individual poker chips, and the stack of the poker chips, that is called granum. So one individual poker chip, in quotes, is a thylakoid, and the stack of them are called granum. Okay, so then the next piece is going to be the stroma. So remember I told you on the previous, the stroma with an R is the jelly in the room. So you can see the space in between all of the stacks, all of those granum or all those thylakoids. There is free-floating semi-fluid gel that is going to fill that room. Okay, so that's why I call it the jelly in the room. This is the space where all of the enzymes are going to be present to help with the chemical reactions. This is equivalent to cytoplasm within a cell. And this is also equivalent to the matrix within mitochondria. There's a nice little bigger picture of each of them. If you look over here, this is a TEM, transmission electron micrograph. And so that is the type of microscopes that can go ahead and send electrons through structures, and you can see the details. And so you can see all of the stacks of those thylakoids, and the stacks, like I said, are called granum. So right here, what we're looking at is we are looking at our photosynthesis formula. And so it is what molecules are used in the formula, and also what energy are used in the formula in order to make the process of photosynthesis happen. So if you recall from your previous chapters, everything on this side of the reaction that goes into the reaction is called the reactant. And actually, I'm going to back this up. I'm going to go ahead and erase. Okay, so these guys are the reactants. I'm just going to put an R, make it nice, okay? R for reactants. There we go. It says plus light energy. Now, yes, you do have to have light energy. Usually, my preference is to put the light energy on the arrow. That is my preference. But, you know, different sources will present them in a different way. And the reason why I'm presenting it like this is because the energy is what is entering and facilitating the reaction and being used in the reaction, but it is not a reactant. It is not matter that is going into the reaction, and it's not matter that is coming out of the reaction. So let's see what comes out of the reaction. And on this side of the arrow, you have products. I'm not going to draw it out. I'm just going to put a P for products. So you can go ahead and write that out. So your reactants that enter into your photosynthesis, what we're looking at is we are looking at the gross equation. And I'm not saying gross equation in the fact that we think it's yucky. I'm just saying it's not the net equation. So you have 6 CO2s that are going in. And those 6 CO2s, along with 12 waters, are going to go into the reaction. Light energy is going to be used in the reaction in order to break the covalent bonds and break those covalent bonds and redistribute electrons. Okay. And when it does that, these atoms are going to get new partners. And so the products are going to be glucose molecule, C6H12O6, 6 oxygen molecules, and 6 water molecules. So if you recall, I told you that this is the gross equation. That means there's a net equation. So look over here. You have 6 waters that are coming out. What if I were to minus 6 waters over here? I'm not going to draw H2O. I'm just going to go ahead and mark it off, right? Then I have no waters in the net equation. And then over here, if I put 6 minus 6 waters here, then I end up with 6 waters left. Boom, boom. So you will see your net equation reading 6 CO2s plus 6 H2Os in the presence of light energy or solar energy is going to yield C6H12O6, which is glucose, and 6 O2s as waste. So what I just told you was the net equation. So rather than me writing it out, I will just go ahead and highlight the net. Oh, I forgot to change my color of my highlight. Silly me. Oh man. I'm working with my Apple. It's a lot different than when I'm in class. Darn it. Let's just draw that one again. Okay. So I say minus 6 on this side. And you got a net of 6 H2Os. And then you have minus 6 H2Os on this side. And so they cancel each other out, giving you the net equation. And so your net equation, let's go ahead and go to highlighter, change pen color to yellow. 6 CO2s plus 6 H2Os in the presence of solar energy. Remember, I put the arrow up under there. I'll refix that. Is going to yield glucose, C6H12O6, and 6 oxygens. Okay. So if you recall, in your chapter 2, I showed you how to make sure that those equations are balanced, meaning the amount of energy, the amount of matter going in an equation is going to be the same amount of matter coming out of an equation. Let me draw that arrow just like such. There we go. So, for example, let me go ahead and show you here. There are 6 carbons, right? And that's it on this side of the arrow. Over here, 6 carbons, and that's it on this side of the arrow. That matches. Over here, there are 2 oxygens in 1 CO2 molecule, but there are 6 CO2 molecules. Well, if there's 2 oxygens in 1 O2 and CO2 molecule, 6 times 2 is 12. So there's 12 oxygens so far. Let's see here. There's 1 oxygen in H2O, but there's 6 H2Os. So that's 6. So 6 oxygens here, 12 oxygens here. Don't forget that. And then over here, you got 6 oxygens, right? In 1 O2 molecule, you got 2 oxygens, but you got 6 O2 molecules. So 6 times 2 is 12. So 12 plus 6, that's 18. So we have 18 and 18 oxygens here. And then, of course, you want to do your H's. And so in 1 H2O molecule, you're going to have 2 hydrogens. Well, you have 6 H2O molecules. So 6 times 2 is 12. So you have 12 hydrogens on this side of the reaction. And if you look over here, it says it right here, you got 12 hydrogens, and that's it. So the number of matter going in the reaction is the same as the matter coming out of the reaction. And the reason why we don't include light energy necessarily, I always put it on the arrow, usually not with the plus. Like I said, different sources are different, is because light is not matter. It is the energy that is being put into the reaction. Alrighty. If you like a clean version, refer to your book. They have a clean net version for you. I'm sure we'll see one here pretty soon too. So this is going to start talking about the splitting of water. So your chloroplasts are going to split water. That's why you water your plants. If you don't water your plants are going to die. And the reason why your plants need water is because they don't really need the water as much as they need the electrons in the water. So what your plants do is they split apart the water molecule in order to remove the electrons from that water molecule. And those plants are going to excite those electrons. And how are they going to excite them? Because you put your plant out in the sun and the sun excites those electrons. And when those electrons become excited, those electrons are going to have a high energy value and eventually end up in a sugar molecule. And then that sugar molecule will have a high energy value because of the excited high energy electrons. And the result, as I showed you, is the release of oxygen. So this is the premise of splitting water. This right here is to show you which of the atoms go to which of the results. So here are your reactants, and then here are your products. So I kind of already went over that when I was counting them out for you. I did it with the net equation, not with the gross equation. And so your photosynthesis is going to reverse the direction of the electron flow in compared to respiration, cellular respiration. So here, this is really important that you hear me, okay? Photosynthesis is not the exact reverse of cellular respiration. Photosynthesis is not the exact same thing in reverse. Number one, photosynthesis, well, not even number one. I mean, this is the whole point. This is one and only. Photosynthesis uses a different energy. It uses solar energy. And cellular respiration uses glucose energy. Okay, so they're not the exact same thing in reverse. But when you look in terms of electrons, the electron flow is going to be in reverse. So in photosynthesis, your electrons come in on your water and leave on your sugar. In cellular respiration, the electrons come in on their sugar and leave out with the water. So that's what that means. And so we've already talked about oxidation and reduction in class. So photosynthesis is also a redox process, oxidation and reduction. Okay, so they just give it a short name, redox reaction. And so basically that means oxidize, when a molecule is being oxidized, electrons are being removed, i.e., the water gets oxidized, electrons are removed. When a molecule is getting reduced, it is receiving electrons. Well, the CO2 that is uptaken by the plant pores is going to receive electrons in order to build a larger molecule called sugar. And so your photosynthesis is an endergonic process. Ender, meaning it absorbs energy. Endergonic process. So this is showing you oxidation versus reduction, and this is also showing you that net formula that I went over. And so when you look here, your carbon, your CO2 molecule is going to get reduced, okay? Meaning it receives electrons. In order for the CO2 to get larger into a bigger molecule, to become sugar, it needs to get more electrons. Why? Because you can't hook on more hydrogens and carbons and oxygens if you don't have more electrons to help it hook together. So the CO2 is reduced, meaning it's an electron stealer. So it's an electron, self-esteem should be reduced. I don't know if you guys remember that from lecture, but there you go. And your H2O got oxidized, meaning during that splitting of water, right, the H2O let go of electrons. Electrons were removed from H2O. So H2O gave away electrons, and it went from being a larger molecule to a smaller molecule, O2. So it comes in as a larger molecule, releases electrons, and it leaves out with just a couple of O's hanging out together. Okay, so things you need to know, the stages of photosynthesis. Your photosynthesis consists of light reactions. Your light reactions are going to be that photo part, meaning it says it in the name, light reaction. Sunlight or some type of light, we have artificial light that we use, but light is used in this part of the reaction. And so the Calvin cycle is, considered the synthesis part. They used to call it the dark reaction. It's not really correct. It's kind of an old school terminology, but really it's the synthesis part of the photosynthesis reaction. So in the photo part, the energy is obtained from the sun. And in the Calvin cycle, that energy is put to use in order to synthesize the sugar. Okay, so your light reactions in the thylakoids, what they do is they split water. They release O2. We kind of already saw that a little bit. They will reduce. What does reduce mean? It means this NADP plus. So it looks a little familiar. In your cellular respiration, you saw something called NADPH. Well, this is a similar molecule, but it is not the same. So NADP plus means, man, he's got a positive charge. Wouldn't he love to have some electrons? Because of his positive charge, he's attracted to electrons or attracting electrons. Well, he's going to get reduced, meaning he's going to take electrons. And when he takes those electrons, that'll help him to hook on an H and then become a new molecule called NADPH. Boom. So in this process also, ATP is generated from the molecule of ADP, and the process is called photophosphorylation. And the purpose of this ATP is not for the kind of cellular work that we would be using it, that the mitochondria would make. Yes, ATP is used for cellular work, but specifically the ATP that is made in photophosphorylation, that is going to be used in the synthesis part of the Calvin cycle. So that ATP gets used to make the sugar. So we're going to go through all of the different stages. This really talks about some of the stages to get started. I'm going to go ahead and show you the big picture right here. And we're going to go ahead and pause right here and then talk about that. Okay, we are back. So we left off on looking at the interior of our chloroplasts, because this is going to be the site in which photosynthesis takes place. So if you look at your structures of your chloroplasts, we've already talked about some of the structures when we introduced them. And so what do we have here? Let's go ahead and laser pointer. On the outside, remember we have two membranes. We have an inner membrane and an outer membrane of the chloroplast, okay? Your quote unquote poker chips, these little green flat circles. Each one is called a thylakoid, and then another thylakoid, and another thylakoid, and then so forth. And the stack of the thylakoids is called granum or grana. Grana is one stack. Well, grana is several stacks, and granum is just one stack. And so this is where the site of the light reaction is going to take place. And if you recall, we also had something called stroma. Stroma was the jelly in the room. So all of the semi-fluid gel in the room is called stroma. And so with that, that is where the Calvin cycle is going to take place. So if you recall, we had ourselves a formula of, let me go ahead and get my beautiful pen here. Actually, I'm just going to back it up so you can see the formula. Go ahead and write it down. A little bit difficulties. I'm working an old thing and it's not working on my iPad, so it's all good. C6H12. I'm sorry, I'm going backwards. Haha, just joking. 6 CO2s plus 6 H2Os in the presence of light energy. Okay, I'm going to put on the arrow solar energy. I already told you how I felt about that. I'm going to put solar E on that arrow. So CO2 comes in through the pores, your water comes up through the roots, solar energy, you put your plant out in the sun, it is going to make C6H12O6, which is glucose, and the result, the byproduct waste, is going to be 6 oxygens. So go ahead and refer back to this as you need to. When you look here, what do you see? You see water is coming in, so that fulfills the H2O component. So water is going to come in and it is going to be split. When I say split, I mean the H's and the O's, the H's and the O are going to separate. And when they do that, they're going to leave electrons here inside the thylakoid. And a couple of oxygens will get together and they will leave outside of the chloroplast as O2. So oxygen is actually a waste in this light reaction. Well, in order to get that water to split, well, you have to put in solar energy. You have to put in light in order to get that to split. Okay, so that is part of the light reaction. There's some very important reactants that need to be used in a light reaction. And one reactant is called NADP+. We talked about that. The NADP+, this is an electron carrier, meaning, remember I told you it's got a plus, a proton, a positive charge. So that means it is in search of electrons. So he's an electron carrier molecule. So he's going to go into the light reaction, pick up some of those electrons that the water left behind, and it'll pick up one of those H's too, and it will leave as this molecule NADPH. So this is considered a high energy molecule. And the reason why the NADPH is a high energy molecule is because the light energized the electrons in here. When the water dropped off electrons, the light energized them. And then the NADP plus came in and got high energy electrons and then grabbed an H and left out as an NADPH molecule. So this is considered a high potential energy molecule. It's got a lot of potential energy. Other components that are coming into the light reaction are ADP, adenosine diphosphate, a low potential energy molecule, and an inorganic phosphate, just a phosphate group that they call it inorganic because it's not attached to the ATP yet. So it is not participating in an organic molecular form at this point. So it is going to come in ADP plus a P, and then in the light reaction, it is going to, through the process of photophosphorylation, if you remember previous slides, going to be made into a high energy molecule called ATP. Boom. So both of these molecules, ATP, high energy molecule, and NADPH, high potential energy molecule, are both going to go to the Calvin cycle. And the Calvin cycle is located in the stroma of the chloroplast. So it is in the Calvin cycle that the CO2 is going to be used. So CO2 enters into the Calvin cycle, and those carbons from the CO2 are going to be used to build sugar. And so the CO2 comes in, actually 3 CO2s total. They're not giving you the totals on here, but I'll just go ahead and tell you now. 3 CO2s, well, it's actually 6 total. Actually, that's why I'm not giving you the totals. Just know, lots of CO2 is going to come in at this point. I'll break it down for you as you get a little further. They're going to come in, they're going to get used in the Calvin cycle, okay? ATP is the energy that is going to drive the Calvin cycle. Just like your car needs gas to drive, well, the Calvin cycle needs ATP to drive. Then your NADPH, if you recall, I called this molecule high potential energy molecule, but I also called it an electron carrying molecule because it's carrying electrons from the light reaction. And so it's going to carry the electrons to the Calvin cycle, and it's going to drop off those high potential energy high potential energy electrons in the Calvin cycle. And they are going to be sequestered in a very controlled fashion to build this molecule here, CH2O. This right here is the building block for a sugar. Because if you recall, a glucose is a C6, H12, O6. So that building block of CH2O times that by 6, you got yourself a glucose. All right, so the important take home, you absolutely need to know what molecules are being used in which cycle. Water is being split in the light reaction. Waste comes out as oxygen from the light reaction, okay? NADPH leaves the light reaction. ATP leaves the light reaction. They enter into the Calvin cycle where they will be used. CO2 is used in the Calvin cycle, okay? And your sugar is going to be a result from that Calvin cycle. Now, when the ATP give off all their energy to drive the Calvin cycle, they're going to leave as an unconnected molecule and low energy, an ADP plus a P. When your NADPH drop off electrons and leave some hydrogens here also to help build the sugar, it's going to leave as an NADP plus, where it's going to go back to the light reaction and get some highly energized electrons and then repeat the process. And your ADP plus your P are going to go back to the light reaction, and they are going to get rebuilt via the process of photophosphorylation, which is just an enzyme that is going to be used in the reaction in order to attach the ATP to ADP to the P. All righty, so big take homes. Know your structures: thylakoids, granum, stroma. Okay, so this is kind of repetitive. Your chloroplasts are solar-powered chemical factories. And so what is light anyway is the next question. So light is a form of an electromagnetic energy. It's also called electromagnetic radiation. And like other electromagnetic energy, light is going to travel in rhythmic waves. And so those waves, they're going to be measured in wavelengths. And what wavelengths are the distance between the crest of the waves. So your book has a picture on wavelengths. I kind of like the internet picture better, so I'll just go find one. So depending on the distance of the crest of the wavelengths, that actually determines the electromagnetic energy of the wave. And so let's do a couple more slides and then I'm gonna go get that picture like we talked about. The electromagnetic spectrum is going to be the entire range of electromagnetic energy that comes from the sun. Okay, and so the part that is used in photosynthesis is going to be visible light. It is a very small part of all of the energy that comes from the sun. And so visible light is going to be used by those plants. And it is basically, it's also going to produce, not basically, but it's also going to produce the colors that we see. And so that's why they call it visible light. And so light also behaves as though it consists of discrete particles. And those discrete particles are referred to as photons. I have some excellent videos that illustrate this. And so they're on my YouTube, and I believe I've also put them on your D2L. And it shows the how the photons are going to work in photosynthesis. So this is representative of the wavelengths, but I would rather give you the one that actually shows the wavelengths. So let's go find that. Let's see, light wavelengths. Let's see if I can find my perfect images. This is my go-to one right here. Let's go ahead and open this in new tab. So as you look at this is showing you visible light and all of the energy that comes from the sun. And so in this small section here, just in this tiny little section here, when you open it up, this is visible light. And so it is going to, this is the light that we see. So when you see purple, what you're really seeing is a reflection of the wavelength around here. When you see more of a kind of a blue purple, that is the wavelength of the light here, and so forth, and going through. So when we see light, when we see color, we are seeing the reflection of those wavelengths. So let's look up here. When you look at the peaks, see how far these peaks are. So at this point, this has a lower amount of energy, but not the lowest amount of energy. As you get further in between the peaks, that is going to be less energy. And so examples of those wavelengths, radio waves, radar, TV, AM, and then also if you look at other ones, other charts, you'll see microwaves embedded in here, infrared waves. A lot of animals see infrared, and so they are able to see those energy wavelengths. And if you see this, ultraviolet light, so UV light, this is the one where you get sunburn, UVA, UVB. Let's see. What do we have to throw in there? I don't really have any great ones that are specific, but if you can imagine the energy that causes you a sunburn, well, it's greater than visible light. And so this is also the energy that can cause skin cancer too. So the smaller you get between peaks, the greater amount of energy that is in those wavelengths. And the more energy in the wavelengths, the more possibility to cause cellular damage through causing DNA damage and mutation. Well, DNA damage leading to possible mutations. Alrighty, so X-rays, the peaks are getting closer. Gamma rays, very, very, very high energy. Those peaks are really close together. So all of this together, this is all of the energy that comes from the sun. And so it is only the small amount here, the visible light, that is used for photosynthesis. And we're not talking about it just yet, but I'm going to go ahead, and since I have this up, if you see here, this is the section that is greenish. It's kind of green, yellow in that section. So when we see green plants, what's happening is the pigments inside the plants are not absorbing this section of the light wavelength. Instead, they're transmitting that light, and they're also reflecting it. So when they reflect it, that's why it comes back to us as this color. That's why we see it as this color. And I just realized I went through that whole thing and did not share with you. So sorry. Let's try again. I'm not going to re-record any of this. So let's see, new share. My apologies. Alrighty. I'll just go over it real quick again. What do we got? We got our peaks between here and here, because that was the whole point of me pulling this up. This little small amount, this is visible light from this section to this section. If you open it up, whenever you see this purple, you are actually seeing reflecting of that wavelength. And I was telling you, when you look at plants and you see green, it's because they are reflecting this wavelengths back to you, and that's why you see it. It is not absorbing these wavelengths, and that is the nature of the pigments, most of the pigments within the chloroplasts. They will reflect these, and we'll talk about that here in a moment. So over here, you have your radio waves, very low energy, a lot of space between the peaks. There will be microwaves embedded in here, and you have your infrared waves. If you look over here is your ultraviolet waves. These are the ones that can cause cellular damage, sunburn and such that I was saying. There's your X-rays. Obviously, you know that causes mutation and cellular damage. And then your gamma rays. So as your waves get closer together in peaks, you have increasing energy. Okay, let's go back to our PowerPoint now. Sorry about that, friends. So I started mentioning pigments. Hang on for the moment. Let me make sure I got this PowerPoint showing correctly. Perfect. I started mentioning pigments. Remember, I told you that there is a pigment called chlorophyll, and that chlorophyll is the pigment in the chloroplast. It is why the chloroplast looks green. Okay, so the pigments are substances that absorb visible light, and different pigments. So plants have more pigments than just chlorophyll. Different plants have different pigments. Some of them have a bunch of different types, some of them have small different types, and it changes between species, the ratio of the types. And so different pigments are going to absorb different wavelengths. And so your wavelengths that are not absorbed, if they are not absorbed, they're going to be reflected or transmitted. And that's why when they're reflected, that's why we see plants as green, because we are seeing what the chlorophyll is not absorbing, but it is reflecting it. So that's what this is right here. Your chlorophyll that is inside, it's embedded inside the thylakoids. Remember our anatomy of our plant? This is the chloroplast, and these are the thylakoids. The chlorophyll is on the membrane of the thylakoids, and there's lots of them. And that's why when you look at your chloroplasts, you see them as so green. So this is just more on light and reflecting. We already talked about absorbing. So what we didn't talk about was chlorophyll A is the main molecule of most photosynthesis. Okay. But because chlorophyll A works best at a smaller range, we need other molecules too. So here is your chlorophyll A, right here. Here's your chlorophyll B. That is another type of chlorophyll. They are so very similar. They are literally just one functional group different when you look at the molecule. And then this is showing another pigment called a carotenoid. And that one absorbs different spectrum range. So when you're looking at the spectrum range that it's absorbing, so here is your light wavelength, your light spectrum. And your chlorophyll A is it's going to absorb greatest right around here, and then it drops off in its absorption rate to just about hardly anything. Doesn't absorb, picks up back again right around here. Another nice little peak and then down again. So your chlorophyll B is going to widen that gap of absorption. So it widens the gap and then drops off, and then starts absorbing around here. Your carotenoids absorb less of the energy because this is what is being absorbed is the light energy. So less light energy, but it absorbs it over a greater spectrum and then drops off. Okay, so if you go ahead and superimpose all of these together, you end up with this action spectrum here. And as you can see, it's exactly what I told you. Right around this area, you don't have the green wavelengths being absorbed. So that is why it is reflected. And that is why when we look at green plants, we see green. Now, depending on what kind of plant it is, it might have different ones. There are lots of different color plants out there, and you will see that they are reflecting other colors and not the green. So we're going to go ahead and scoot through. I'll let you go ahead and read about the bacteria and how they figured out how bacteria also is, they are attracted to specific wavelengths, but that's not something that we're going to cover. So those two that I just told you about, your chlorophyll B and your chlorophyll, your chlorophyll B and your carotenoids, they are called accessory pigments. Like accessories, you can't go out the house without your accessories. And so, boom, carotenoids. And what they do, like I told you, they will broaden the spectrum for photosynthesis, right? And then also the carotenoids, they will absorb excess light. And by absorbing that excess light energy, without this present, that chlorophyll might get damaged. So they help to keep the chlorophyll from getting damaged. They absorb a lot of the excess energy. Like I told you, is just one functional group different. So this is the molecule of chlorophyll, and chlorophyll A, if there's a CH3 there, it's chlorophyll A. But if there's a CHO there, then it's chlorophyll B. And that is the difference between the two molecules. So when the pigment absorbs light, it is going to go from a ground state to an excited state. And that excited state is unstable. And so when the electrons are excited and they need to release that energy, what they end up doing is they have to release that energy to something. And so they'll try to fall down to a ground state. When we say ground state, it means a lower state of excitement. Photons will be given off. And when photons are given off, if there's not other molecules there to receive those photons, instead a glow will take place. And that glow is called fluorescence. So when you have this in different organisms, that fluorescence, and they glow in the dark. We've talked about this with the dinoflagellates in the Vieques, Puerto Rico. We had a little discussion about that in class. So this is just an example. It's kind of a model on energy state. So this is the level of energy of electron, okay? From the lowest state of energy to the highest state of energy. Then, let me put my little laser pointer. Here is a chlorophyll molecule. So an electron is embedded in the chlorophyll molecule. This is considered a ground state as far as the levels of energy. And we're just saying that this is a low level of energy. When this electron is just gotten to the chlorophyll, it's just doing its thing. How did it get there? Well, water came in, got split, released an electron into the chlorophyll molecule. And it wasn't until sunlight sent photons, choom, 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 ching, and excited that electron. Well, when it excited that electron raised its energy state to a higher state of energy. Okay, so if you can envision this, you've done this a lot. Have you ever taken your socks and just walked across a carpet? What are you doing? You are exciting those electrons. Same thing if you take a balloon and rub a balloon on your hair, you're exciting those electrons. So you've done this before. So high energy, this is a high state of energy, electron being highly energized. So they can't just stay here. They have to release their energy. So those electrons are going to return back down to a ground state. But as they do that, they are going to release heat as a byproduct. We've talked about heat as a byproduct of energy transformations. And they're going to release photons. Why? Because the photons came from somewhere. And those photons being released are going to cause that chlorophyll to fluorescence. And so literally this is chlorophyll and it is glowing. It's hard to tell. It looks like it's green, but if you're under a black light, it's glowing. Okay. So the meat and the potatoes. At this point, we have talked about the overall big picture of photosynthesis. We've talked about the nature of light, because without understanding the nature of light, you can't understand how photosynthesis works, because it has to do with electrons and energy. So this next piece is the specifics of the photosynthesis systems. And so the first system is going to be when the light is harvesting. And so we have light harvesting complexes. And as much as I just want to read through this, because really I don't, I just want to open up a picture and talk about it. So feel free to read through that yourself if you like. All righty, so what do we have here? This right here, as you see, look at that. We got phospholipids and phospholipids. They're phospholipid bilayers. Huh, that looks very much like a cell membrane, doesn't it? Well, that's because it is very similar to a cell membrane, but what we're looking at is the thylakoid membrane. Remember the thylakoid membrane? We talked about the thylakoid being the quote poker chips. Well, that's what the membrane of the thylakoid is made out of. And so when you look at out here in this area on the outside of the thylakoid membrane, that is the stroma, the semi-fluid gel in the chloroplast. And then on the inside of the thylakoid membrane, they call that thylakoid space. So it's important to know that. So let me draw just a cute little picture real quick, so that way you can reference. I probably should grab myself another color. There we go. I'm just going to draw it up like this. Here's my poker chip. Not really a poker chip, is it? It's called a thylakoid. Boom. Okay. So all of this green is the thylakoid membrane. Okay. On the inside is thylakoid space. Go back to black. Because it's so hard to write with my little tiny mouse, on here I'm just going to put TS. And TS is for thylakoid space, right? And then on the outside here is stroma. Probably work out stroma. There we go. Okay. So what I'm going to do is take my little green pointer, pen color, change it back to green, and I'm just going to remind you that this green, oh, I didn't mean to hit photosystem like that. That was just me being clumsy. Try again. Okay. There you go. That's what that is. That is your thylakoid membrane. So I just wanted to draw it like we had seen it before. That way you can recognize that it's the quote unquote poker chip. Okay. And then so in that thylakoid, embedded in the thylakoid membrane is going to be your proteins. And you've seen this before. You've seen this illustrated with our cell membrane, where your cell membrane has proteins? Well, it's going to be the same. And so let's go ahead and draw a protein on the membrane. Boom. All right, now that you got the gist of what we're looking at, now I'm going to refer to it on here. Okay, so this protein that is embedded in the thylakoid membrane is called a photosystem. And so your photosystem is going to be the site in which photosynthesis takes place. So all of these green dots, those are chlorophyll pigment molecules. Okay, and so this one that looks like a double chlorophyll molecule, it's a specialized pair of chlorophyll molecules. I won't make you remember, depending on which photosystem, it has a different name. So I won't make you remember what the name is. We could just call it a specialized chlorophyll molecule. It's good. So these are going to have electrons already in them, okay, just sitting around waiting. And then this is going to have electrons in them also. The photons are going to come from the sun. Choom, 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 choom. What they're going to do is they're going to excite the electron in this chlorophyll molecule, in this chlorophyll pigment. And then that's going to play past the excitement and past the excitement. So just like where I told you about rubbing your feet on the carpet with socks, and then you go and you touch somebody and pass that excitement, and then so forth and so forth. And so they just continue passing that excitement and building up energy that already was given from the sun. And this final accepting chlorophyll molecule, they're going to get ultra excited. And when the electrons in this molecule gets ultra excited, it raises these electrons to a higher state of energy. And when these electrons are released because of that higher state of energy, there's going to be a molecule waiting, just catch them. So think about baseball when the catcher catches the ball that is being thrown by the pitcher is getting really excited, and it comes at a high energy pace, and there's a catcher there just ready to catch it. All righty. So this is just what it looks like molecular wise. And so they call that a primary electron acceptor. Just kind of scooting through. Remember, I'd rather look at pictures than read words. I will tell you, there are two different types of photosystems, because I just told you that there's types of photosystems. There's photosystem 2, and then there's photosystem 1. And no, it's not out of order. Photosystem 2 comes first. Photosystem 1 comes second. It is thought that photosystem 2 was discovered first. Wait, no, I said it wrong. It's thought that photosystem 1 was discovered first, but then they found out there was another one, and then that actually comes in order. So let's just watch. We're going to also talk about linear electron flow. Oh, I hate reading. Matter of fact, yeah, I'll go through this model. It's a quick model. Like I told you, here are the chlorophyll molecules, pigment molecules. The electrons in them are going to get excited by the sun, the photons, choom, 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 choom. They play past the excitement to the electrons that are in each of the pigments. And then the final accepting chlorophyll molecules has electrons in here. It's going to raise those electrons to a higher state of excitement, and those electrons are going to be released, and the primary electron acceptor is going to take them. Well, when it takes them, they have to go somewhere. He can't just sit on them. So I'm going to come back to that. So he is going to send them in this direction. Well, now, since these electrons are gone, these need to be replenished. That's where your water comes in. Your water will split, release electrons to that specialized chlorophyll molecule, and replenish those electrons. A couple of oxygen atoms will go off to become molecular oxygen. All right, so let's see where these electrons went. These electrons are going to travel down an electron transport chain. If you recall, in a cellular respiration lecture, we went over electron transport chain. It's the same thing. It's a series of enzymes that are on the membrane that are going to help to slowly remove energy from those electrons. And when it removes the energy from the electrons, it builds up that energy to produce ATP. And it does it almost identically to the way it does it in cellular respiration. So just think this orange is the path of electrons. It is going to travel down the electron transport chain. This is just a model form. It's not showing you on the actual membrane. And then once they travel the electron transport chain, they release a bunch of their energy to the electron transport chain in order to make the ATP. Now these electrons are tired and they need to settle in to a new specialized chlorophyll molecule in photosystem 1. So I forgot to tell you, this was photosystem 2, and this one's photosystem 1. So what's going to happen in photosystem 1, once again, light is going to excite the electrons in these chlorophyll. They play past the excitement and build it up until that excitement is passed on to the final chlorophyll molecules, raising the electrons here that came from the electron transport chain to a higher level of excitement. Primary acceptor molecule is going to take them and send them down a little mini electron transport chain. I'm going to be honest with you, when we are discussing electron transport chain, we are talking about this, okay? This is just a little mini one. Now, it's usually not our main reference one. All right, so ferredoxin, FD. Ferredoxin is an enzyme that is going to receive the electrons from this primary acceptor molecule. It is going to take those electrons and help build the NADPH molecule. It does that by this enzyme, NADP plus reductase. Ferredoxin will pass those electrons to the NADP plus reductase, and then it will grab some H's that are floating around, and it'll become NADPH. So the ATP that is made in the electron transport chain and the NADPH that is made at the end of photosystem 1, those get sent to the Calvin cycle. This is just another model showing energy levels. This is showing cyclic versus linear. I'll back it up. When we're talking about linear, we're talking about the linear flow of electrons. Electrons come in on the water, they go through, they go through, they go through. Electrons go through, go through, go through. Electrons leave on the NADPH. There, at the same time that there's a linear process, there's also a cyclic process where some electrons will just go back and go to the big participating electron transport chain round and round and round in order to make high amounts of ATP. They do that because you need a lot more ATP than you do NADPH. Okay. We're going to take a little pause. Okay, we are back. Excuse me. Just trying to make sure I got the right screen share. All right, perfect. So you're probably wondering, what does mitochondria have to do with this? Well, if you recall, we had talked about the electron transport chain being both in the mitochondria. We had discussed that in our last lecture, and on the chloroplast. So this is just to show you the similarities and then also the differences. So when you're looking at the electron transport chain, where it faces in the mitochondria. So here is your mitochondria, and here's your inner folds called the cristae, if you recall. So that's the dark gray. And here is your light gray. Your light gray is the matrix, and the matrix, remember, that is equivalent to cytoplasm. Excuse me. And then the dark gray, that's the inner membrane space. It's between the outer membrane and the inner membrane. So let's just look at our picture here. We have that inner membrane space. And then if you see here, you got the matrix. Now, when you compare it, look at the chloroplast. So just ignore this side now. Now look at the chloroplast side. So when you look at the thylakoid, you see the thylakoid space is dark gray. And so here is the thylakoid space. And then this represents the thylakoid membrane. And the light gray is representing the stroma. So if you recall, I told you that the stroma is equivalent to cytoplasm and the matrix is equivalent to cytoplasm. So these are great connection points for you to realize where the structures are facing, because on both of them, you have an electron transport chain, which is a series of enzymes that are going to support the removal of energy as the electrons are being transported across the chain. And then you also have what is called your ATP synthase. Remember, I told you your ATP synthase is the biggest, baddest enzyme that there is. And the function of the ATP synthase is to produce large amounts of ATP. So what we're trying to show you here is ATP is going to be made, as far as the chloroplast is concerned, in the stroma. And when you're comparing it on this side, ATP is going to be made, as far as the mitochondria is concerned, in the matrix. So it's really easy because both of those are equivalent to cytoplasm. Well, guess what? That's where all the enzymes are. In that semi-fluid gel, that is where all the reactions take place. So that's an easy remember. And that's what this shows. It shows the similarities between the two organelles and which way the ATP synthesis face. Okay, so we've done a lot of discussing of the light reaction. We've just looked at it in model form. This is another look at the same concept, but now we have put it on the thylakoid membrane, because really that's what we want to do, is we want to connect it to that organelle that is working. Okay, so here's your thylakoid membrane. There you go. I don't know why I have a pointer and a thing. That's weird. Hang on. I just want a laser pointer, not a laser pointer and an arrow. Let's try that one again. Okay. I see the laser pointer is kind of small. Let's see if we can get a bigger one. No? How about a different color? No. That's right. We tried. Okay. So here's your thylakoid membrane. Where did my laser pointer go? There we go. Here is your thylakoid membrane going all around. So on your thylakoid membrane, you're going to have those proteins, this big protein called photosystem 2. You're going to have these enzymes embedded. All these enzymes together make up the electron transport chain. And then you have this big protein, and it is called photosystem 1. Then you have these enzymes, ferredoxin and NADP plus reductase. And when you look down here, you have the biggest, baddest enzyme of them all, of the electron transport chain, and is the ATP synthase. Okay, so let's go ahead and go. Let's do some light reaction of the photosynthesis. Because at this point, we haven't made any sugar. By the time we get done with this, we will have made some high energy molecules, but they won't be sugar. We'll have to go to the next system for that one. So if you recall, you have lots of electrons in the chlorophyll molecules that are in photosystem 2. Those electrons get excited by light energy. They play past the excitement till they raise to a higher state of excitement, releasing the high energized electrons to an electron acceptor molecule. And that electron acceptor molecule is then going to send those electrons down the electron transport chain. Okay, we're going to come back here. Water, because you watered your plant, is going to release electrons into that specialized chlorophyll, and they're going to replenish those electrons. When that water breaks apart, hydrogens are going to float around in here. So you can see got some hydrogens. It should draw more hydrogens. I think that would be nice for illustration. And then oxygens are going to float around in here. And a couple of oxygens get together and they're going to make an O2 molecule, molecular oxygen. All right, that'll be waste. Let's go back to our electrons that that started on the electron transport chain. Those electrons are going to travel the electron transport chain, giving up a piece of energy, giving up a little bit more energy. And every time they give up a little bit more energy to, like, for example, this enzyme, he's a carrier enzyme, plastiquinone, he's going to help to pump hydrogens across the membrane. And those electrons travel more and hydrogens will pump across the membrane. And then a new carrier molecule will take these electrons to the next photosystem and let them settle into the chlorophyll, specialized chlorophyll molecule, where once again, light photons will energize those electrons, play past the energy, build it up, excite the electrons that are here, raising them to a higher energy level where they're going to be received by the acceptor molecule. That acceptor molecule is then going to give them to ferredoxin. Ferredoxin is going to pass them on to NADP plus reductase. This ACE enzyme is going to help NADP plus some H's and use those electrons to quote, glue them together. But maybe if we want to be official, we would say covalently bond them to make NADPH. That NADPH is going to get sent to the Calvin cycle to be used. All righty, so let's go ahead and go back in here. Your hydrogens, if you recall, were being pumped across the membrane by using that electron energy. And when they get pumped across the membrane, they start building up a high hydrogen concentration in here. And if you recall from diffusion, number one, you don't like a high concentration in an area because they're going to want to diffuse to a lower concentration space. And then also, these are all protons. So all those pluses, they just start getting kind of anxious and they just want to get away from each other. So they're going to diffuse down their concentration gradient. And as they diffuse down their concentration gradient, they're going to go through this specialized enzyme called the ATP synthase. You already saw this when we did our cellular respiration. Your ADP is going to be sitting there waiting. And as those hydrogens diffuse down the ATP synthase, it's going to cause a shape change in this enzyme. Just shape change in the enzyme. And when it has that perfect shape, it's going to encourage these guys to get together to become ATP. And ATP is going to get sent to the Calvin cycle to be used in the Calvin cycle. So what I've explained to you is the light reaction. The light reaction happens on and within the thylakoids. And the products of the light reaction are going to be sent to the Calvin cycle because we have not produced any sugar yet. It is here in the Calvin cycle that the sugar gets produced. So your Calvin cycle has 3 phases: carbon fixation, reduction, and regeneration of the CO2 acceptor, RUBP. RUBP is the molecule that is called the CO2 acceptor. Hang on, there we go. All righty, so what we're doing is we're playing count the carbons. When I taught you cellular respiration, I played count the carbons with you. I showed you how a 6-carbon molecule, which was that glucose, would break down and completely oxidize by the time it went through cellular respiration. Well, this is going to be just the opposite. This is going to be counting the carbons and eventually building up that glucose molecule. But really in this, you want to count the carbons within the cycle. And they call it a cycle because of the same thing with the citric acid cycle. It is going to have the exact same molecules at every single stage, and then it is going to it's going to cycle around again. So I'm just going to make this big, and then maybe I'll back it back off again. Let me see. There we go. So when you look at this, number one, your Calvin cycle is going to go around 3 times. 1, 2, 3. Okay. Well, look at that. It's going to input 3 carbon dioxides, one at a time. So one CO2 goes in, it'll cycle around, go through all this process. Another one goes in, it'll cycle around, go through all the process. Another one goes in, and it'll cycle around and go through the process. All righty. So with that, and then it'll actually do that again with another 3 molecules in order to produce a full glucose. But this is just showing what it takes to make a half a glucose. So in this, as the CO2 comes into the cycle, there's a special enzyme called ribisco. Remember I told you that enzymes usually end in ACE? Well, if you look up the full name for ribisco, it is ACE. But you know what? They just call it ribisco, but it does have ACE. So ribisco is the enzyme that is going to receive those CO2 molecules. And what it does, as it receives those CO2 molecules, there's already this molecule waiting around. It is called RUBP, ribulose biphosphate. And if you count it, let's count it. How many carbons? 1, 2, 3, 4, 5. It has 5 carbons. And so the RUBP is going to be sitting here with the ribisco waiting for a CO2 to come in. CO2 comes in and it's going to attach that 1 carbon to this 5 carbon molecule. Let's see if it does a good job. Let's count them out. 1, 2, 3, 4, 5, 6. There you go. So it did its job, and this is very short-lived. As quickly as it comes together, it's going to go ahead and break apart. So let's count these. There are 3 RUBPs, and you're going to have 3 incoming CO2s to make 3 6-carbon molecules, right? 5 carbon plus 1 carbon is 6 carbon. So there are 3 6-carbon molecules, and then they will all break in half. Click, 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 click. Now there's going to be 6 3-carbon molecules. Count them all up, you got the same amount of carbons. If you don't believe me, let's try. 1, 2, 3, 4, 5, 6 times 3 is 18. Come on over here. 1, 2, 3 times 6 is 18. So we did not get rid of any carbons. We just reconfigured the molecular where the bonds were. All righty. Now this has a special name called 3-phosphoglycerate. Now, if you were in my class sitting in a chair taking a test, you would have to write this out for me. But guess what? You don't. But 3-phosphoglycerate also has a little mini word called 3P, an abbreviation, 3PG. And I've also seen it as PGA, but most of the time it's 3PG. All righty, so that 3-phosphoglycerate is going to receive, each one of them, because there's 6 of them, they are all going to receive an ATP molecule, and that ATP molecule are going to drop off a phosphate. Look at that. There's 6 ATP molecules. They're going to drop off a phosphate. Boop. Oh, look at that. There was no phosphate here before. Now there is. So they each received a phosphate, and then 6 of those, once they dropped out the phosphate, they left as ADP because they're no longer triphosphate. They left a phosphate behind. They leave as diphosphate. And they will just go back to the light reaction to get reconfigured. All righty. I know this is a mouthful. Let's see what's next. Once they phosphorylate and drop off a phosphate, they become a new molecule, and they become 1,3-biphosphoglycerate. But I want you to see something fun. Look at this. Remember, it was a 3-PG. Then watch this 3-PG. And it dropped off a phosphate. Well, it changed into a new molecule called BPG. Can you see that? I put that in black. So 3-PG, 3-phosphoglycerate, after phosphorylation changed into BPG, which is 1,3-biphosphoglycerate. Yeah, that's how I got through school. All right, so then this BPG, be like boy, is going to receive electrons from the NADPH molecules that come in. Where did they come in from? They came in from the light reaction. And so 6 NADPH molecules, one for each of these, is going to, they're going to drop off electrons. When they drop off electrons, guess what? This phosphate just falls off. It doesn't need to be there anymore. And now we have a new molecule, and that new molecule is called G3P. So I'm going to go ahead and highlight. Well, yeah, I do want to highlight that one. It's called G3P. So we went from 3-PG to BPG to G3P. Yeah, say that quick. Well, there's 6 of them, 6 G3Ps, glyceraldehyde 3-phosphate. So your glyceraldehyde 3-phosphates, they are partial sugar molecules. So one of those is going to leave. One G3P is going to leave. He's a partial sugar molecule. And 5 of them are going to move forward and continue on in the reaction. So 5 G3Ps move forward, and those 5 G3Ps are going to be used to regenerate the original acceptor molecule. So let's count them out. In one molecule, there are 3 carbons, but there are 5 of those molecules. So 5 times 3 is 15. All righty. So in order to get these to reconfigure, we're going to have to add in some energy. And that's where 3 more ATP will come in and drop off phosphates and then help to reconfigure all of these bonds and carbons. Because remember, this is a 3-carbon molecule, 1, 2, 3, and there are 5 of them. So 5 times 3 is 15. Drop off some energy, break some bonds, make some bonds. Now you have a new set of molecules. 1, 2, 3, 4, 5 carbons, but there are 3 of them. 3 times 5 is 15. All we did was take these and reconfigure bonds, break some and make some, and then now we have these molecules. And these molecules are the acceptor molecules that are going to accept the CO2 molecule. So after you go through 3 times, okay, 3 times is going to make you 1 G3P. Well, we need to add up, because this is a 1, 2, 3 carbon molecule. How many carbons in glucose? C6. There's 6 carbons in glucose, meaning this all has to happen another set of times. That way we can get another one of these. When you get another one of these, then you can put them together to build a glucose molecule. See, 3 carbons, 1, 2, 3, and then a whole new set of these come again, and it goes again, and then you get 4, 5, 6, and then you can have yourself a sugar. So the big take home, Calvin cycle takes place in the stroma of the chloroplast. The light is not used in the Calvin cycle. CO2 enters into the Calvin cycle. The products of the light reaction, which were ATP and NADPH, those products become the reactants of the Calvin cycle. The final product of the Calvin cycle is a partial sugar molecule called G3P, and those partial sugar molecules will add up to make a glucose. No light is used in this reaction. Okay, so here's what I suggest. Now, I'm not going to go through all of this stuff. You can go ahead and read about C4 plants, C3 plants. Don't go into too much detail. Just know that these ones are in extreme condition. CAM plants and C4 plants, extreme conditions. You can go ahead and read about them on your own. But here's what I want you to do. I'm sorry if I'm scooting around. I'm going to slide back. Because this can be overwhelming, and there's a lot of stuff, and it's a mouthful. Trust me, I've just spent about an hour and a half to 2 hours spitting it out. Start here. Start right here. Learn the overall big picture. Learn the structures. Learn what goes in and where it goes in, what comes out and where did it come out from, what goes in and where it goes in, and what comes out and where did it come out from, what the products are and where they go, what the once they break down, what leaves there and goes back in here. So these are the products of these reactions, and they become the reactants. So I said it backwards. These are the products of this reaction, the light reaction. These products become the reactants in here. Then they are going to produce products that become the reactants in here. So just understanding where these are made and where they go and what leaves, and then it gets remade. So this is your starting point to look at the big picture, to understand where everything is and what is going on there. Then look at the details of what is going on in a photosystem and the structures. Then apply that photosystem. Sorry, guys, but it is what it is. Then apply the photosystem here and here, because this is photosystem 2, and this is photosystem 1. So apply that concept to where the photosystems are. I'm not going to go over it again. You can just rewind. Then see what the products are, ATP and ADPH, see where they go. So if you take it in steps, it becomes less overwhelming. Start with that big picture. So if this was more confusing than it was helpful, I apologize. I don't make biology, I just teach it. I would have loved to be with you in class, but I probably would have just said the same thing. There are some very great videos for you to see. Let me go ahead and stop. Actually, no, I didn't want to stop share. Let's go to this desktop. I don't know where I'm going. Let's just stop sharing for a second. There we go. I want to go to Google, not that Google. Not that Google. This one. I know I got a lot of Googles open. That's the problem. Don't even know what I have where. Okay, so if this becomes too confusing, doesn't mean don't watch it, but maybe before pulling your hair out, you take yourself over here into your, I'm looking at 112 here, but I'm sorry, 2H1 here. But if you see in your lecture, you have some videos that will probably say everything I said in 5 to 10 minutes. So light reaction video, it's right here. Beautiful. This one shows the photons. It really sums it up. So nice. And then we have ourselves total photosynthesis, including light reaction, Calvin cycle, and electron transport chain. And then we also have ourselves Calvin cycle just by itself. So I hope you enjoy. Thank you very much.